Some believe that high-throughput screening is kind of black box. So you throw a sample in and it spits out the perfect hits. And automation is misunderstood as a synonymous with simplicity. So the reality is high-throughput screening is highly complex. It requires very carefully assay development and validation, and also a deep understanding of the biology, precise robot programming, and sophisticated data analysis. So the system must be calibrated, maintained, and constantly also monitored. And that could also lead maybe to suboptimal decisions when the team believes it's just pushing a button. They underestimate the need for highly specialized personnel.
Welcome back to part 2 with Sebastian Blum from Beckman Coulter. We're continuing our conversation on mastering early-stage bioprocess development. We're talking about practical guidance on choosing between shake flasks, benchtop reactors, and high-throughput platforms, and understanding when each approach makes sense for your specific application. We're also covering the common pitfalls that trip up even experienced teams and how to avoid them. Thanks to Beckman Coulter for making this episode possible. If you want to make smarter screening decisions and save months of troubleshooting, this episode is for you. Let's dive back in. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brühlmann, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. What would you tell a startup founder who says, well, I'm interested in the BioLector because the system looks great. We can test a lot of things. We can move quickly, as often startups want. But what I'm hearing also from your answer, Sebastian, is that you need some technical background to be able to use the system correctly. Would you advise against or for, or under what conditions would you say, no, this makes sense? You will get the most out of using a BioLector?
Normally, when a customer comes to us or we are approaching customer, we are very intensively talking about their applications. And we just had, just as an example, one customer, it's an academic customer, who wanted to do strictly anaerobic cultivations with a very low OD. So they started an OD at 0 point something and wanted to induce at 0 point something. When we recognized this, we thought, okay, that was out of specifications. For example, our BioLector resolute from OD1 up to, let's say, sky is the limit, because of the scattered light. And there we tried to understand how important that is for the customer. And when he told us that this is essential for his process, we said, okay, then the BioLector is choice for him. So this can happen, but this is on an application-based level. The nuanced scripting is not very often used, especially not in academia, interestingly. So very often it's done in industry where you try to, for example, combine different feeding mode in one run so that you can mimic the bigger fermenters, which have also different feeding mode, like an exponential feed that is then going to a linear feed or a constant feed. This is possible with this nuanced scripting, and that does not happen too often. But of course, if customers are asking for this, as mentioned before, the solution is there. They cannot handle the Lua scripting. We can do that. But this is normally not a showstopper.
So what are the scenarios in which it makes the most sense to use the BioLector?
I would say everything that has to do with process development, finding right parameter combinations, like media, like pH, induction profiling, and where also potentially is a need for flexibility on different microorganisms, like for example, CDMOs. They need to be very flexible. And for them, it's important to have a system that is not dedicated for one single microorganism or one single application, but they want to serve or support their customers with different microorganisms. I would say the BioLector is a very good fit, especially because we have the possibility to upgrade our modules to the BioLector depending on the application. For example, a customer can start with a simple system that is a batch system, upgrade this one with a microfluidic system, and this microfluidic system allows the customer then to do, for example, feeding and pH control, as mentioned before, of 32 wells then. And if he sees I have a need of an anaerobic application because a customer or project calls for it, then he can upgrade the system with anaerobic module to be able to do feeding, pH control in 32 experiments, also strictly anaerobic. And if he says then, okay, now I need some offline analytics, for example, you can also put the whole system into a Biomech i5 or i7 to have the full advantages of the automation, for example. So I would say if a customer asks for flexibility and is screening a lot of parameters and trying to find the best conditions, that's where the BioLector is located.
So that means it's particularly suited for early stage screening, correct?
Yes, exactly. This is 99% of our systems placed in the upstream development, early R&D screening, indeed.
Speaking of automation and Biomech, where do you see companies typically place the BioLector in their workflow?
Like mentioned before, we have the upstream development, downstream development, and finish. Typically, we are seeing the system only in upstream development. So this is where you're doing all your strain development, trying to find the best media and setting up those initial culture conditions to get the best growth and productivity. So in the process, this is the upstream development fermentation.
What are the most common misconceptions you see in scientists wanting to acquire the BioLector or have it already in-house? Because from what we've discussed so far, it's a powerful tool. You can do a lot of things, but sometimes it's also about managing expectations and helping people use the technology best. What are the most misconceptions scientists have about high-throughput screening systems in general?
Some believe that high-throughput screening is kind of black box. So you throw a sample in and it spits out the perfect hits. And automation is misunderstood as a synonymous with simplicity. So the reality is high-throughput screening is highly complex. It requires very carefully assay development and validation, and also a deep understanding of the biology, precise robot programming, and sophisticated data analysis. So the system must be calibrated, maintained, and constantly also monitored. And that could also lead maybe to suboptimal decisions when the team believes it's just pushing a button. They underestimate the need for highly specialized personnel. So they might invest too little in assay development for bioinformatics expertise. The result is often poorly designed assays, unreliable data, high error rates, so false positive or negatives, and ultimately the loss of valuable time and resources because the results cannot be interpreted or reproduced at all. So the black box, I think, is something, it's not that simple. It's not a black box, you put things in and you just get the perfect hit out.
This is very well said, and I've done a lot of deep well plates experiments in my career, and knowing what you're doing and understanding the process side of things and analytics are just very important. So thank you for highlighting this, Sebastian. Looking ahead with now more and more technology on our end, on the robotic side, on the automation side, and with AI, just to throw in a buzzword, I'm curious about what is your perspective of the future? How do you see early stage bioprocess development evolving?
What I see is more automation and miniaturization. There's a clear tendency regarding into this direction. So we are going to see even higher throughput, more sophisticated microbioreactor systems, and a lot of more integrations of robots for end-to-end automation workflows. So from media preparation all the way to sample analysis. AI, yes, the buzzword. So you see it on every conference at the moment as well. The huge amounts of data that high-throughput screening generates will increasingly be used with artificial intelligence and machine learning for predictive modeling. really fast identification of optimal conditions, a much deeper understanding of the mechanisms at play. It's going to move beyond just finding what works to understanding why it works. So I see there's a lot of potential of AI. I see also that tendency is going into this direction. What I see as well is that, especially in the bigger industries, we see more and more digital twins and process simulations. So expect to develop more robust in silico models, basically digital twins, bioprocesses, and that will let scientists simulate thousands of scenarios virtually before they even do a physical experiment, which will speed up optimization even more. I think that's still a little bit the future, but I see that customers are talking to me about this idea or this big goal. I still think there are some skills which will always remain critical. The core bioprocess engineering principles, so you still need that fundamental understanding of microbial physiology, cell biology, fluid dynamics, mass transfer, and reaction kinetics. The tools might change, but those underlying biological and engineering principles are constant, in my opinion. And of course, you need to have critical thinking and problem-solving attitude. So the ability to really critically evaluate data, figure out the root causes of problems, and design robust experiments, that will always be at the heart of successful development, in my opinion.
Before we wrap up, Sebastian, what burning question haven't I asked that you're eager to share with our biotech community?
Well, I have to say, David, the questions you are asking are quite good. And I've also watched your podcast in the past and was also surprised about the nice spectrum that you can show there. So in my opinion, everything was said here. I really appreciate your questions and also your podcast. So thanks a lot for me being here and being interviewed by you.
Sure. Thank you very much for the feedback. Great to have direct feedback from listeners.
Yeah.
By the way, if you have feedback about the podcast Smart Biotech Scientists, please leave a review. This means the world to me. As we're wrapping up, Sebastian, what is the most important takeaway from our conversation?
In my opinion, it is to think about where to put in high-throughput screening tools in your process and to evaluate carefully the needs and the advantages of this for the customer's application. I hope that I a little bit highlighted here the possibilities that customers do have by using BioLector for this purpose. Still, I encourage people to stay curious and to stay critical and to choose the right tool for the right job.
Well, this has been fantastic, Sebastian. Thank you very much for helping us better understand the key success principles for early stage screening, for sharing all your insights. Where can people get a hold of you and learn more about the BioLector?
The Beckman.com homepage will reveal a lot of information about BioLector, including publication lists and also application notes that are available there. A gallery where you can see the system, how to use it, a kind of small training part. So I think if somebody is interested in getting in touch with BioLector, the Beckman.com homepage would be perfect. And in Europe, if there's an interest in Europe, normally I'm aware of this as well. And at some point of time, me as a market developing manager will also show up here.
Excellent. Great, Sebastian. I will leave the info in the show notes. And once again, thank you very much, Sebastian, for being on the show today.
David, it was a pleasure. Thank you.
Thanks for joining us today for this deep dive into bioprocess screening strategy. Remember, the right early stage decisions save months of troubleshooting downstream. If today's conversation gave you actionable insights for your own development work, please leave us a review on Apple Podcasts or whatever platform you're listening on. It truly makes a difference. Thank you so much for your feedback. I love hearing from you. And until next time, keep doing biotech the smart way. All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.